cold in here. Oh, hey, it's 1 o'clock. I guess it's time to get started. How many? Doesn't seem like we have a lot of students yet. Let me see how many of you are here. Let me get my laser here. Where is everybody? There's only 6 students. Everybody's going to view it later. Is that the deal? I guess. I think it's time to get started. Yeah, 1 o'clock. Well, if you're opting to view this recorded, I guess that's fine. You don't need to see me live for anything, but today is Tuesday, January 26th. My office is kind of loud. I'm on the 3rd floor of the science building, and they're doing work, so you can hear some knocking around in the background on occasion. Anyway, but today's our 2nd regular lab. Last week, remember, we did the animal tissues. You know, some people joining late. That's okay. So today, we're doing that 2nd chapter out of your atlas. It's called the Perpetuation of Life, and basically what it is talking about cell division and development. And traditionally what I do for this lab is have my students look at 3 different slides. One slide to study mitosis, 2nd slide to study meiosis, and then a 3rd slide to study early development. And you'll talk about, I'm sure, these topics in lecture, but you get a look at them in lab. And so what's the first thing we're going to look at? Well, to study mitosis, which is the nuclear division of somatic cells, basically where cells divide, you have one cell divides into 2, right? And each of the cells are genetically identical with each other, and of course the parent cell. So they're basically clones of one another. But there's different stages that take place in the development. You might go into more detail in lecture, but in lab, using light microscopy, that you're limited to how much detail you can see. And so basically what we do is look at the basic stages of mitosis that take place. Now, where would you view this? Well, mitosis occurs anytime the cells are dividing for one reason or another. If it's a multiplying bacteria or protozoans or whatever, that's how they reproduce. Or in growth, you're going from a one cell stage, like a zygote, it's going to divide to form a 2 cell, all the way up to, like, your body has about 100 trillion cells in it, 100 trillion. But you started off as 1 cell. So how do you go from 1 cell to 100 trillion cells? Well, the cells actually divide, and they divide again, but they multiply. So they multiply. And so they do that under mitosis, or actually mitosis is the special events that take place in dividing the cell. In the contents of the cell, of course, you've got chromosomes, the units of heredity. And so every organism has a set number of chromosomes. And so to ensure that each cell has the correct number of chromosomes, they have to divide a special way. And so to study this, what we've done is taken like fertilized fish eggs, and we study them. And we've taken the eggs from this species of fish called a whitefish, which is real common used in the fish hatcheries. And so fertilized fish eggs are readily available. And so the egg, when it's first fertilized, it's one cell big. Like the egg, unfertilized egg is a cell, one cell. It might because it's a big cell, but it's still a cell. And then once it's fertilized, its scale is going to start dividing and growing. And so, the fish egg, the whitefish egg is tiny, it's about this big, but it divides and divides again, divides. And so if you want to study the stages of mitosis, you look at it in the area that's readily undergoing cell division. And so that's why we're looking at this whitefish egg. And it forms a cluster of cells called a blastula. We're going to talk about the different stages later. But so that's what we're doing. And so this field of view, what you're looking at is a fertilized fish egg. It's actually gone from a zygote up to this blastula stage. And what you see is cells everywhere. And so here's one, and here's this nucleus, here's another one, and this nucleus. And most of your cells just kind of look like normal animal cells, all right? And you can see characteristics of the cell. This is the nucleus. This is the nuclear membrane. Here's the cytoplasm. And inside the nucleus is nucleoplasm, the contents of the nucleus. All right, and so forth. Now, you don't see chromosomes or anything like that most of the time. The only time you're going to see chromosomes is if the cell is going to divide. And so most of these cells, if they just look like the normal animal cell with a intact cell membrane, intact nuclear membrane, the nucleus, nucleoplasm, it's not dividing. It's not undergoing mitosis. If it's not undergoing mitosis, that stage in the life history of the cell is called interphase. And so most of these cells that you see, with the exception of this one, let's ignore this one for right now, most of these cells are in interphase. And so that's kind of what you think of. Now, here's a close-up of one cell in interphase. Now, the word interphase means between stages, or like if the cell's not dividing, it's an interphase. So this is like between division stages. A long time ago, like when I was a student, they used to call this interphase the resting stage. Well, it's not really resting. Cells never really rest. They're always doing something, okay? Interphase is actually busy. The cell is manufacturing proteins, doing this, that, and the other. And if it's going to divide, it's going to replicate all the DNA inside of there. But only when it's going to divide is the DNA in the nucleoplasm. At this point, it's just called chromatin. The chromatin would condense and form visible chromosomes. But if you can't see visible chromosomes and you see the nuclear membrane is intact, you just identify this stage in the life history of the cell as interphase. Okay, so it's not really a stage of mitosis, it's a stage in the life history of the cell. So what comes next? Well, if the cell is going to divide, the chromosomes are going to form from chromatin, okay? And that's what you see right here. Look at this cell, this kind of different stain, it's just a different slide altogether. But I'm showing you this one because this is a whitefish blastula cell that's in prophase. Prophase is the first stage of mitosis. And I recognize that for a couple of reasons. One, like where the nucleus is, this is not a nucleus, okay? I know it's right here in the center where the nucleus should be, but I recognize it as not being a nucleus because I don't see a nuclear envelope or a nuclear membrane. So if there's no nuclear membrane, of course, there's no nucleus. But in the area where the nucleus was, you got this kind of grainy looking material. It's kind of looks like, I don't know, like a hairball or something like a cat coughed up or something. And what that is actually the chromatin condensing into chromosomes. And this happens during prophase. Those chromosomes really haven't done anything. other than just appear, okay? And they're in preparation, getting in preparation to divide, but they haven't divided yet. So this is what prophase looks like. Okay, so here's prophase. So what comes after prophase? Well, the second stage of mitosis is called metaphase. Okay, meta means like next, like what's coming next. Well, prophase, all the chromosomes appear, but in metaphase, the chromosomes actually connect to these little bitty fibers. I don't know if you kind of see that there's some little like stringy looking fibers descending down from this side of the cell, and then the opposite side, you got some more. These are called spindle fibers. The spindle fibers develop from opposite poles of the cell, and they descend down and they hook up to these chromosomes. And the chromosomes at this stage are actually a chromosome and a copy. It's like here's a chromosome and its copy, and together they're kind of stuck together. They're called sister chromatids, and they're stuck together in the center part, which is called the centromere. On either side of the centromere, you have spindle fibers connecting, and what they're going to do is they're going to connect there, but then they're going to pull those sister chromatids apart. So you can end up with two daughter cells with a sister chromatid in each. And so if this was a human cell, you would have 46 of them doing that. So you'd have 46 going towards each cell, and once they're divided, they're just considered, sister chromatids are just considered chromosomes. But in metaphase, this is what it looks like. They're before they're pulled apart, they're all kind of lined up in the middle here. I think M for metaphase and M for middle, or like the equator. If you think of this as like a globe, this is like the North Pole and the South Pole and the equator in the middle. All the chromosomes line up along the equator, right there in the middle. And they're held in place by these spindle fibers, which in turn, once everything's in the middle, they're going to pull and shorten and pull these sister chromatids apart. That hasn't happened yet, but you'll see, and it's really easy to recognize. But this, from prophase, you go to metaphase, and this is what it looks like. So metaphase to anaphase is the third stage of mitosis. And here I show you like three different stages of anaphase, or three different, because remember, this is all fluid, it's happening, one right after the other. It's not real choppy like this, and so it's kind of gradual. So you get some other cells in different stages, like right here, this looks like a metaphase, and then they're going to be pulled apart. And right here, you can kind of see that they're barely being pulled apart. So you're going to end up with two sets of chromosomes. Right here, they're called sister chromatids being separated. They're being pulled apart right here, and here they be pulled. This set is being pulled about a little bit more. You kind of see some space occurring between the two, and then definitely right here, you can see big space, and you see two different sets of chromosomes headed towards opposite poles. So anytime you see the separation taking place, that's anaphase, okay? This is like early, middle, and late anaphase, because, it starts out like this, and then looks like this, and then this, and this. Now, this is just a great slide. Don't expect, like if you're looking at a slide, don't expect to find them always together like this. This is just dumb luck where I found this. And, but this is a beautiful one. That's why I wanted to share it with you. Early, middle, and late anaphase. And I know it's anaphase again because I can see that there's separation between the two sets of sister chromatids. But at this, I would call these, they're chromatids until the cell is completely separated. But they're going to be, once they're in opposite cells, chromosomes. And so what comes next? Well, the final stage of mitosis. Up until this point, we're just separating the chromosomes. But, the ultimate goal is to form two new daughter cells, okay? Each daughter cell has to have, the correct number of chromosomes, and each daughter cell has to have its share of cytoplasm. And so what happens in the last stage of mitosis, This is the cell right here, but notice the cell has this indentation in it. See that? It's like pinched in. That indentation right there is called cleavage, the cleavage furrow. So in the last stage of mitosis, which is called telophase, like tele means like terminal, like at the end, the last stage. And you recognize telophase, the presence of that cleavage furrow. It starts off as like a little dent and it gets deeper and deeper and deeper and deeper until it completely pinches through and forms two new cells. But as long as they're connected, it's still considered one cell. It's not two new cells until you have two new cell membranes intact. But this is still considered one cell. It's still in mitosis. And this is the last stage. I recognize it because you can see the cleavage furrow here and here. And here, of course, are the two sets of sister chromatids separated. And it's eventually going to be, you know, one complete cell here and here, but it's still connected. So this is telophase. And, you know, you could have early, middle, and late telophase based on how deep that cleavage furrow is. If it's just a little bit, early telophase, a little bit more, you know, middle telophase. And if it's like almost all the way connected or all the way separated, say late telophase. But if you can see a little connection between them, it still would be telophase. So this, you know, what is this? Is this early, middle, or late? Mid to late, you know, because it's almost it's almost completely through, but it's definitely telophase. Okay, that's how you recognize telophase. And that's it. I guess that's it. And so once they're separated, the telophase would segue to basically interphase. It end up look like, you know, the cells would look like this, where you know, you get a cell membrane and there's a reappearance of the nucleus. Because I can see this is a nucleus, nuclear membrane. See this, these cells right here look different than this. You get to dark chromosomes. No chromosomes in here. This is just you know, nucleoplasm with the chromatin in there, and so no visible chromosomes. So this is this one and this one. Interphase. So this is what these after telophase become two daughter cells in interphase. Okay? Does that make sense? So those are the stages of mitosis. Do y'all have any questions about that? You think you could recognize this again? Like if I showed you, like on your practical, so how are you gonna ask us on your exam? Well, let me show you a slide like this and ask you, hey, name this stage of mitosis, right? And I might show you this one and say, okay, name this stage of mitosis. And now I want you to recognize this as anaphase. You know, name this stage. You should recognize this as metaphase. Can y'all handle that? I think you can. It's not too bad. No questions? Okay. Well, there you go. That's mitosis. Now, mitosis is a form of nuclear division. When I say nuclear division, the nucleus of a cell, not the nucleus of an atom. And I know there's a nucleus in chemistry and there's a nucleus in biology. In chemistry, you're talking about atoms, like of elements. Okay. In biology, a nucleus is the organelle in the cytoplasm of a cell. Okay. And that's the part that contains all the genetic material in the form of DNA. All right. So nuclear division. We have a name for nuclear division. It's called karyokinesis, and there's two forms of karyokinesis: mitosis and meiosis. What's the difference in mitosis and meiosis? Well, mitosis is the karyokinesis in the formation of somatic cells. Somatic cells are cells in the body. Soma means body. Somatic means of the body. So somatic cells are the cells of the body, all the cells in the body other than the cells that form sex cells or gametes. And so what do you call the karyokinesis of somatic cells? Well, that's meiosis. That's what meiosis is. Meiosis is the karyokinesis in the formation of somatic cells, like forming sperm or eggs. So why do you have to have a special form of, well, the wrong way, karyokinesis for that? Well, what is so special about gametes or sex cells? Well, sex cells, sex, S-E-X, in biology, it only means one thing. It means genders. So the terms gender and sex are synonymous in biology class. It's kind of funny because I just saw somebody arguing this on Facebook about gender and sex and whatever, you know, and they're talking about, I guess it was, I don't want to get too political or any crap like that, but I'm just going to let you know, your biology professor is telling you that there's only two genders, there's only two sexes. I think the argument was that, oh no, there could be many genders. There's only two sexes, but there can be, that's stupid. No, I don't know who made this crap up. No, there's no such thing. Gender, sex is the same thing, and your sex and your gender are determined at conception. When you're conceived, you know, you're going to automatically be one sex or the other, one gender or the other. You know, what you grow up to mimic, that, you know, whatever, you know, that's up to you. But biologically, you're going to be one sex or the other. Okay, well, that's a different argument. I don't want to talk about that. But what allows that to happen, your gametes, of course, you know, you get a male gamete, sperm cell, and female gametes, the egg. And this is how we reproduce. And those eggs or sperm, the chromosomes are different. Okay, they only have half the number. Remember, in the somatic, in mitosis, the cells that are produced have the same number of chromosomes. They're genetically identical to one another. They're clones of one another. Remember I said that? Well, in gamete formation, or sperm formation, or egg formation, what happens is that the cells, the gametes, have half the number of chromosomes. Okay, so a sperm cell or an egg, human, would only have 23 chromosomes opposed to a somatic cell, which would have 46 chromosomes. Same thing for an egg. A human egg would only have 23 chromosomes. Why is that? Well, 23 and 23 together makes 46, and that's how you start, right? A human zygote has 23 and 23 or 46 chromosomes. You got half of them from your mom, half of them from your dad. And from that one cell, it divides by mitosis and goes from a zygote all the way up to 100 trillion cells. And each one of your cells in your body has 46 chromosomes, that began with that same zygote that, you know, with half of your dad and half your mom. Okay, and there's different chromosomes in there that do different things. And one pair is to determine your sex, and so that happens at conception. You know, you're either gonna have an X and a Y chromosome or two X chromosomes. With two X chromosomes, you can be a female. If you have X and a Y chromosome, you're a male. Period. Okay. So, how does that happen? Well, there's a special form of karyokinesis. That's meiosis. So meiosis ensures that the daughter cells have half the number. Okay. Now you'll see the detail of all this in lecture. What's actually happening in different stages, and they got names for the stages and the types of cells that are formed and so on and so forth. When lab, really limited to what we can see in light microscopy, okay? And in studying meiosis, you're really limited, again, what you can see in the laboratory because it doesn't always happen the exact same way in males and females as far as the stages. I mean, you can see all the stages in both sexes, but in one place, if you want to see all the stages in one situation or one region of the body, you can only do that in males. So what am I talking about? Well, everybody knows that the sperm is produced by the male's testes, and the eggs are formed by the girl's ovaries, right? Well, all the cell division that takes place obviously takes place in the ovaries and the testes. Well, that's kind of true, that all takes place in the male's testes, but in the female, it begins in the ovaries, But then the egg that's developing is ovulated, okay? And when it's ovulated, it's not finished with meiosis, all right? So it doesn't really finish the meiosis stages until after it's ovulated. So you can't just go to the ovary and see all the stages of meiosis. And so that's why we just look at the testis. The testis to study, the stages of meiosis. You can see them all in here. So it starts from a primordial diploid cell. Diploid just means that it has all the chromosomes, both sets, okay? And it ends up with a haploid gamete. Haploid means has half the number of chromosomes. So you start with 46 and you end up with cells that have 23, okay? That's for humans. Different animals have different numbers of chromosomes. And so whatever. So to study meiosis, again, in our lab, what we do is you got to go to the gonad. Here's the gonad. We're looking at a male gonad. Our subject is a rat. So we're looking at rat testes. And you're in the testes, inside the testes, you actually have the structures where meiosis is taking place, and that's in the seminiferous tubules. So this is a section through a rat testicle, and then these are the tubules in the testes where the cells are actually dividing. And so each one of these little round things that you see in there, these little purple donuts or whatever, see they got little holes in them, they're hollow. That collectively are referred to as seminiferous tubules. So this is one seminiferous tubule, that's another seminiferous tubule. They're everywhere. The testes is jam-packed full. Human testes exact same way. But we're looking at rat testes in our lab, okay? Because we couldn't get any volunteers male human volunteers to donate their testes for us to study this. Mice and rats don't complain too much, so we take their testes and slice them and look inside of them. Okay, so there we go. Seminiferous tubules to look at meiosis. There we go. Here's a zoomed in seminiferous tubule. Okay, you can see it's hollow in the center, and then you see the wall of it is made up of a bunch of cells. Okay, and these cells are undergoing nuclear division, or under karyokinesis, right? But reduction division. And we're talking about reduction division, you're talking about reducing the number of chromosomes. And that's meiosis. Meiosis, reduction division, same thing. Or the karyokinesis and the formation of sex cells. And it begins out here in the perimeter. These cells divide and they move towards the lumen until they're in the center here, until you have fully formed sperm cells. And you can kind of see this. You can see in the middle, see this along these stringy, kind of wispy looking things. Those are the tails of the sperm, or the flagella. The flagella is like a whip-like structure that's on a cell that allows it to be mobile. Okay, so that's what you see. You see the tails of the sperm, and the heads of the sperm are kind of tucked away up here. But you really have to zoom in to see that kind of detail. And so what we're going to do in lab is identify specific cells at different stages of meiosis, beginning out here and working our way towards the middle. And so here we go. Look at this. This is zoomed in. One seminiferous tubule right here. Here's the lumen, the hollow part of the seminiferous tubule, where you can have the sperm cells. And like I said, you begin out here and you work your way towards the middle. I just kind of like pointed out different stages. This first cell you see right here, the little dark one right there, that's called a spermatogonium. And the spermatogonia are the primordial, primitive cells that all little males are born with. And these are the cells that haven't really divided or started the reduction division and so forth. They haven't undergone meiosis yet. So they're just kind of formed by mitosis, and they just make copies of themselves. And this is the only thing that really exists in a male prior to puberty. When he's born, that's what he has. He just has spermatogonium. But when he reaches puberty, what happens, these guys kind of replicate themselves, and every once in a while they're going to replicate and then form a new cell. These big ones right here, see these kind of fat ones that are kind of right next to them, see them right there, these big ones? These are called primary spermatocytes. And we identify these big primary spermatocytes because these are the cells that are destined to undergo meiosis. They're like chosen to undergo this reduction division. Spermatogonia make copies of themselves, spermatogonia, and after puberty, they'll spit out a primary spermatocyte every once in a while. So both the spermatogonia and primary spermatocytes are genetically identical. They both have 2 sets of chromosomes, so they're both diploid, but the primary spermatocytes are going to undergo reduction division. They're going to undergo meiosis, okay? And they're going to lose, eventually produce these haploid cells. And so this is what you start with. And then these guys are going to divide, okay? And what a primary spermatocyte divides into is the first stage of meiosis, okay? Which is called meiosis 1. And what happens in meiosis 1 is the primary spermatocyte splits into 2 and forms 2 secondary spermatocytes. And you can recognize secondary spermatocytes in your slide because they're the most numerous, and they're right here in the middle of the seminiferous tubule. You look at these cells right here, see them? Everywhere. Here, they all kind of look the same. They're all about half the size of this one, okay? These, from here to here, all these cells are secondary spermatocytes, and they all look pretty much the same. They're everywhere, see them? The most numerous ones, and they're all formed from these guys. So this is like a different generation. So these primary spermatocytes here, and then secondary spermatocytes here. And that's what happens in meiosis 1. In meiosis 1, you go from primary spermatocyte to secondary spermatocytes, okay? We're not finished yet. These cells have to divide again. What happens to the chromosomes between primary and secondary spermatocytes, or in meiosis 1, is that all those chromosomes, what they do is they remember, you get 2 sets, well, a set from mom, a set from dad. They're called homologous pairs. What happens in meiosis 1 is the homologous pairs separate into daughter cells. And so that's what you have here is the one set of chromosomes in one secondary spermatocyte and the other set in the other. Well, at the same time, these chromosomes are duplicated, so they exist as sister chromatids. And so the sister chromatids need to separate. And so what happens in meiosis 2 is the sister chromatids will separate. Now, all this detail you're going to learn in lecture. In lab, I just want you to recognize them. You know, I'm not going to ask you, any of that detail. I'm just, in labs, it's all being recognition. I'm going to say, what cell is this? What cell is this? What cell is this? Okay. Well, I'm just kind of letting you know the reason why, okay? What happens in meiosis 2 is the secondary spermatocyte they're going to divide and they're going to form spermatids, okay? And spermatids actually have those separated sister chromatids. And you can recognize spermatids as these nice, round, little dark, just it looks like just a nucleus. It's round little things. Those are spermatids. Now you think, well, we're done. Well, spermatids are immature sperm cells. Okay, they're not mature yet because they don't have little flagella and stuff. So the spermatids actually have to mature and develop a tail and all that kind of stuff. They have to metamorphose. How about that? So they're the transition, and they go from spermatids to spermatogonia, or mature sperm cell. It's easy to recognize because you say, hey, they have a tail on them. So I think I show you that next. Yeah, this is just a narrow slide. You can clearly see these wispy, kind of swirling, looks like a vortex in there. That's created from the flagella of the sperm set, spermatids. And the heads of the sperm, they're not round. They're more oblong. So they look like dark little streaks. Thickened the dark little streaks as the heads of the mature sperm cells. And I think it says sperm heads and something. What does that say? Residual bodies? I'm sorry. I don't know what that says. Anyway, the heads of the sperm are kind of tucked away right here, and the tails are kind of like swirling it down the lumen. So this is what it looks like in a rat. A rat sperm, human sperm. Not exactly alike. All animal sperms are a little bit different, but generally this is what all the sperm in seminiferous tubules would look like. And so there you go. So how's that? That's spermiogenesis, or spermiogenesis is what you call the maturation of the spermatids to the mature sperm cells. So flagellated mature sperm cells, that's what you see right here. It's a result of spermiogenesis. Now spermatogenesis is actually the meiosis in the formation of the spermatids, okay? But going from spermatid to sperm cell is spermiogenesis. That's lecture stuff. Anyway, so that's that. Y'all have any questions about anything yet? Mitosis, meiosis? It's like I said, it's just all recognition. Practice going over the slides. Quiz yourself. Okay, what cell is this? What cell is that? I've got things pointed out for you already. Cover up the answers, whatever. You have access, again, to this PowerPoint. It's on my courses. It's called Perpetuation of Life, PP or something like that, and for PowerPoint, and you'll see it, okay? And of course, you have this recording, or you'll have access to it. There's only 10 of you in the lab. We're supposed to have, how many in this class? So some of you are watching it live, and there's like, so that's 5, well, not too many, probably maybe 20 of you. That's half of you here. So I guess the other half of you watch the recording. That's fine. So what's next? This is our third slide, our third and final slide. And you kind of like this one, it's kind of cute because it's looking at babies. So we know how to cell divide, we know how to make gametes, and so you get the gametes, put the gametes together and make a fertilized egg, and then the fertilized egg develops. So this is starfish development, or the early cleavage stages. Like when a cell divides, it cleaves into two. So early cleavage stages. Now, why do we look at starfish? Well, nobody really cares about starfish babies, you start raising eyebrows if you start, putting humans and fertilized human eggs underneath the microscope, people kind of frown upon that. I'm not going to get political or something, start talking about pro-life versus pro-choice and blah, blah, blah. But I will tell you, I'll just let you know in science, again, life begins at conception. What you do with that life, that's up to you. But I'm not going to tell you the facts. We still get a starfish. These early cleavage stages are identical to what you'd see in a human. So we could be looking at a human, it could be looking at a starfish. Either way, it's the exact same thing. So your early stages look just like this. And so that's why we look at a starfish. Nobody really cares about developing starfish stages. But you look at humans, it gets a little bit too more personal, so we kind of don't do that. So anyway, cleavage stages of a starfish. So look at this. And these are all different individuals, okay? And beginning with a unfertilized egg. And so, now a unfertilized human egg would look a little bit different than this. But this is what the starfish egg looks like before it's fertilized. And you can recognize, it's about this big. This is magnified about 100 times, 100 times underneath the microscope. Here's the cell, here's the nucleus and nucleolus. Okay. Once it's fertilized, the nucleus, like this, it fuses with the nucleus of the sperm cell. So it looks different. And under the microscope, it looks like this. It just kind of looks like it doesn't have a nucleus. It's kind of dark like this. And, but you look like this too. Once and me and everything that's a result of sexual reproduction has a stage in their development where they start as a fertilized egg or a zygote. So this is a one cell zygote. And of course, this is going to go from the zygote all the way up to 100 trillion cells. And it all starts in that first cleavage stage. And I think that's what I show you next. Yeah, right here. Here again is a zygote, and it cleaves and forms the two cell stage. So this is your first developmental stage. Everything has this. Starfish, human, orangutan, kangaroo, bat, earthworm, whatever, doesn't matter. This is a two cell stage. It divided into two, and each one of these cells are, produced by mitosis. So each one is genetically identical to one another. And if it's a human, there'd be 46 chromosomes in here, 46 chromosomes in here. This is a starfish, different number of chromosomes. I don't know. Okay. So easy to identify. Each cell is about half the size of this one, And of course, they're going to continue to divide. And so the two cell stage goes to a four cell stage. See there right here? And again, you look at it, there's four, 1, 2, 3, 4. Each one's about half the size of the original. And from a four cell stage, it's again going to divide again. And it's going to go to an 8 cell stage. And so each one of these divides, and it forms like this cluster like this. They're a little bit smaller, a little bit harder to see, but if you look carefully, you can tell that there's 4 cells here, and then there's 4 kind of in the background. Okay. And so that's, kind of how you see it. And again, it would go, 2, 4, 8, and then 16, and 32, and then 64, all the way up to 100 trillion if this is a human. But I'm not going to sit here and show you all those different stages. As a matter of fact, after 8 cell stage, you really can't count, because the cells are going to get smaller and smaller and smaller, and you really can't count them. And so from 16 to like 64, we have a different name for that stage. That cleavage stage is actually called a morula. See the morula stage? So the morula is kind of like anywhere from 16, 32, 64 cell stage. And it's a cluster of cells. Man, the cells get smaller and smaller and smaller. And so it remains a morula as long as that cluster of cells is solid, okay? If it's not solid, it starts like developing a hollow interior. We call it a blastula. Okay, so you move from morula stage to blastula stage. And a blastula, you can look carefully, you kind of see some hollowness developing on the inside. That's early. And again, the cells divide. As long as there's a hollow center, it's a blastula. But look at this. It's definitely can see a hollow center here, but the cells are so small you can't even make them out anymore. Okay, and so we identify this as much later in development. So this is a late blastula, okay, and this is an early blastula. So that's the order it goes in: morula, early blastula, late blastula. And you went through the exact same stages. Okay, your blastula stage looks a little bit different. That's in mammals. That's it. The early stages. Everything looks alike. And the more closely related you are to a different group of animal, the longer you're going to share the developmental stages. But if the stages start becoming different, they differentiate. In early development, you're more distantly related. So this is about where we segue or separate vertebrates from starfish. Right about here, the blastula stages kind of start looking different. But up to this, we were basically identical. If you think about, all things start off as one cell. Everything looks like one cell, and then 2 cells, 4, 8, whatever. But once you kind of get up there, obviously you kind of start, well, okay, we're not looking the same anymore. So starfish and other members of that phylum, which is called the phylum Echinodermata, all the echinoderms, which would be brittle stars, sea urchins, and things like that. They kind of start looking more sea urchin-like. And then all the chordates, which include all the vertebrates, you know, start looking less and less similar. But then within the vertebrates, you know, certain vertebrates will share the developmental stages longer. So like all the mammals will look more like each other longer than, say, a mammal versus some kind of reptile. You know, reptile will look less like, you know, an amphibian, a fish, and so on and so forth. So anyway, these stages, what comes next? So this would be the latest one, the late blastula. So what comes next? Well, development continues. And so the blastula is the blastula until you see this little growth in the center. You see that little hump that's growing? That's the beginning of basically forming the entrails. You know, so things are happening, you know, the tissues are, you know, start forming, well, the cells are going to start to form primordial tissues. And that's what you see happening here. This is called a gastrula. Gastrula means literally, I have a gut. They have a gut, gastrula. And so that's what's forming is the early primitive entrails, or the guts, you know. And so the little bitty nub right there, that's just an early gastrula, and then it grows and gets longer and fills this interior more and more and more. And we go to the late gastrula. As long as it's forming this little gut-like structure, it's a gastrula. So you go blastula, this will be late blastula, early blastula, late blastula, early gastrula, late gastrula. How about that? Look at that. Now, I'm zooming in and looking at detail of the early gastrula to let you know what's going on in there. So this is the early gastrula. There's that close-up of that little nub right there, all right? And remember I said the cells are now forming primordial tissues, so to speak. The hollow center actually has a name. It's called the blastocele, okay? Because remember in the blastula, there's a hollow. That hollow place is called the blastocele. Well, you recognize a gastrula as a gastrula because it's developing this new tissue that's going to fill that blastocele. The new tissue that's growing on the inside, it's called endoderm because it's growing on the inside. So if this is endoderm on the inside, everything else is on the outside. It's called ectoderm, okay? These are the two primordial tissues that develop first, okay? They actually develop at the same time. As soon as you develop endoderm, everything else is on the outside is ectoderm. So they form at the same time. If you follow the development of these cells, the ectoderm cells versus the development of the endoderm cells, they're going to form specific types of tissues. For example, the ectoderm, the stuff on the outside, it eventually is going to go on to form all the integument. That makes sense, right? Because it's on the outside. Your skin, you know, you had the endoderm too. And your endoderm went on to form all the skin, all the things associated with the skin. And it goes on to form your nervous system as well. The endoderm, on the other hand, is going to go on to form the gut. You know, that's why they call it gastrula, because this is what's forming. The intestines and the digestive tract and all the organs and other things associated with the digestive tract all are descend from or develop from endoderm, okay? So what about the stuff between your skin and your gut? So I'm glad you asked because that's the third embryonic tissue. It's called the mesoderm. Meso means like in the middle. This is the late gastrula, and you recognize the late gastrula as the late gastrula because of the presence of mesoderm, this third embryonic germ layer. All chordates, all echinoderms. Well, there's like two groups of animals in the phylogenetic tree. You have a group of animals that develop from just two embryonic germ layers, endoderm and ectoderm, and then everything else that develops from three embryonic germ layers. They're called diploblastic and triploblastic organisms. Triploblastic just means three germ layers. Diploblastic means two germ layers. Well, chordates, that's our phylum Chordata. And echinoderms, the phylum that includes the starfish, they are both triploblastic. They have three germ layers. And so that's why we're kind of grouped with the starfish. That's why we study starfish. But other really primitive things further down on the phylogenetic tree, they only have two real simple, like jellyfish, really primitive forms of life. They only have two embryonic germ layers. Anyway, so we have three. And so what does this form? Well, mesoderm is going to go on to form this middle kind of layer. And that, if you follow the development, it would go on to form all the muscle in the body and then all the connective tissue. In chordates, the connective tissue would include bone and cartilage and fat and all the tendons and ligaments and things like that. And that's all the connective tissue. And then the muscles, cardiac muscle, smooth muscle, skeletal muscle, all is going to come from this mesoderm. But this is very, very, very early in development. This is like still in the cleavage stages. If this was you, these stages of your development, this is what you would look like right before you're implanting into your mother's endometrium in the uterus. These early stages of cleavage for humans and all mammals for that matter, the cleavage stages occur in the fallopian tube or the oviduct or uterine tube, whatever you want to call it, the tube that connects the ovary to the uterus. Fertilization takes place in the fallopian tube, and it's you go from unfertilized egg to a fertilized egg in the fallopian tube, and then it's traveling down the fallopian tube on the way to the uterus. And as it's traveling, it's dividing. It's undergoing these cleavage stages. And so for humans and all mammals, it goes to a 2-cell stage, 4-cell stage, and it goes to all these early cleavage stages. So right about here, when you have the triploblastic stage, where you have all three germ layers, that's when it attaches to the uterus, and then that's when you're truly pregnant, because you actually have a developing embryo at this stage. These early cleavage stages, called cleavage stages. It's not really an embryo until it's implanted. So there you go. And this is like, it takes about a week for a human. So this is from fertilization to implantation is about 7 days. Okay. So, but this is a starfish, and this is the late gastrula. Okay. This is another kind of views, looking at some other detail of the late gastrula. Now, remember I said you recognize the late gastrula because of the presence of mesoderm, okay? And then mesoderm is forming from that developing little gut thing. Well, the name of it's not the intestines yet. It's not the stomach or anything like that. It's not fully developed. So we call it the primitive gut, or we use the word archenteron. Archenteron literally means primitive enteral or primitive gut. Like archaeology, study of like ancient things. Enteron, like it means enteral. So primitive enteral is a primitive gut. So this whole thing is the primitive gut. So archenteron formation is the development of this primordial gut. Now it's hollow, okay? And so remember right here, the space, that's still the blastocele. And remember I told you the blastocele is being filled with the mesoderm and all that, the developing gut. So this is going to basically fill the space and kind of obliterate the gastrocele. Okay, but again, it itself is hollow. It's like your mouth is hollow, your stomach is hollow, your intestines are hollow. So that hollow space inside of there is called the gastrocele. So don't confuse these two, blastocele, gastrocele. Blastocele is the cavity outside of the archenteron. The gastrocele is the cavity inside the archaeoneron, and that's the same, hollow space inside of your body. The opening to the outside, if you take this individual and rotate it like 90 degrees, it would look like this, okay? See a little hole right there? That's this little hole right here. The hole itself is called a blastopore. So here's a blastopore here, the blastopore here. So why are you identifying this hole? Well, this is another pretty profound thing, is that, remember I said, the more closely related you are to a group of animals, the longer you're going to share these early developmental stages, okay? The further, more distant related you are, the earlier you're gonna diverge in these and share fewer developmental stages. So right here, this is kind of a pivoting point right here in development, because this blastopore in all animals that are developing, this thing is going to go on to form one of two things. Remember I said your archaeoneron is forming your gut, and your gut has two openings to the outside world. One, your mouth, and the other, on the other end, is your anus. And so what is this going to be? Is this blastopore, is that going to be the mouth, or is it going to be the anus? Well, it depends on what kind of animal you're going to be. In two groups of animals, the blastopore is either going to be the anus, or the blastopore is going to be the mouth. And again, this is why we're looking at echinoderms, because only echinoderms and chordates, us and starfish and the like, the blastopore becomes the anus first, okay? And so all other animals, who are the other animals? Well, there's 26 or 28 animal phyla. Echinodermata and chordata are only 2. So the other 26 animal groups, phyla, kingdom, phylum, class, order, family, genus, species, their blastopore becomes the mouth first. Okay, so what are they? Well, maybe not this semester. Maybe in high school you might have learned some animal phyla, but I think they do animal diversity in biology 1202. Maybe you talk a little bit about it in 1201. But for example, segmented worms, Annelida, the annelids, the segmented worms, the flatworms, platyhelminthes, anything sounds familiar to you? Mollusca, the mollusks, these are other animal phyla, and they all go through these same stages, except this is where we kind of divert from them, only echinoderms and chordates, this becomes the anus, and all those other ones becomes the mouth. And so we have two other big groups of animals, all right? The mouth first group and the mouth second group. And we have names for that. The mouth first group, that's called protostomata, which literally means first mouth. The mouth second is deuterostomata, which literally means mouth second. And so we, echinoderms and chordates, are deuterostomes, and all the other animals are protostomes. And again, this is all lecture stuff you'll learn about lecture. In lab, I just simply want you to recognize that is a blastopore, and that's going to be the future anus of a starfish. And there's another view of it. This is the archaeoneron. Of course, this tissue right here would be endoderm. This tissue on the outside, see on the outside, that's ectoderm, etc. This view, you can't see the mesoderm, but the mesoderm would be out here. See how it kind of poking out on the sides? That's the development of the mesoderm. Anyway, there's your leg gastrula. Any questions about that? I know there's a lot going on that slide. You all got that? Think you can identify all that stuff? Okay. What comes next? Well, remember we're developing a starfish, but it's not like bam, just all of a sudden a starfish with 5 legs. As a matter of fact, it doesn't look anything like the adult starfish when it's a larva. And basically what's happening is that archaeoneron is growing and growing and growing and growing until it reaches the other side and forms the mouth. And we got the anus and then the mouth. Once that happens, it becomes the larva, all right? And you have very similar stages. Of course, you don't call it a larval stage. Humans and mammals don't have larvae, you know. We have the embryos, and it's just kind of like your embryonic stage. But anyway, there's a point in your developmental history where all your organs are formed, what they call organogenesis, and then they grow until you're big enough to be born, or whatever. So this is the larval stage, what comes after the late gastrula, okay? And here you go. And so what used to be the blastopore is now the anus, and then you can see right here, there's a little intestine that segues to this kind of roundish sphere hollow. That's the stomach, and then it kind of in the background, it's kind of narrowing. That's the esophagus. It kind of flares open and opens up right here, and then that's the mouth. Okay, these bands on the outside are ciliated bands, and that's how the thing starts moving. It starts moving around, it starts swimming. So it doesn't look anything like the adult starfish. And this is what the larval starfish looks like. It swims around like this until it grows, and it actually has bilateral symmetry. It has, halves, and then it's going to change into radial symmetry, where it has, 5 arms and parts, like a starfish or a brittle star or a sea urchin or a sand dollar would look like. And that's how it goes. Okay. And that's it. I think that's it. La fin. You know what that means? What does that mean? It means the end. Look, that took an hour. Isn't that perfect? Like perfect timing. I think I've done this once or twice before. Okay. But that's it for this week. And like I said, ordinarily, we got 2 more hours. We got an hour for one group to go in the lab and look at this stuff, another hour to do it. But really, I don't see a need to do that. We don't really have to do that unless you really wanted to. But everything you're going to be tested on and you're going to be responsible for is in my PowerPoint. So I think that's it. So do you guys have any questions about anything? I mean, look at this. There's only, let's see how many people are in here. 10, there's only 10 of you in here. So I see your names. No question. There's 2, 4, 6, 8, 9. So there's, and me is 10. So we're good. I don't take it that anybody wants to go to lab for anything. If you want to go, I'll go. But I'm not going to just go in there and sit there by myself for 2 hours. You really don't need to. So I'm not going to show you anything. There's nothing in there that you're going to be testing on that you hadn't already seen. Okay. So, okay, no questions. All right, y'all can handle that. So look at it, practice looking at your slides, your PowerPoints, your recorded whatevers. And any questions about anything, just give me a holler. You can email me, or you can wait till next week, whatever, ask me live if you want. That's it. So, okay. No questions, and I will see you guys next week. All right. Bye.